Thank you so much to the organizers for having me and inviting me to speak here today. I'm really excited to tell you about our manuscript, which describes a modular cell therapy approach for tackling solid tumors. I have a relatively brief presentation here describing the key findings, as well as some key data from another A2 manuscript to highlight the robustness and modularity of the TMOD logic gate system. So as we all know, CAR T-cell therapies have been highly successful in liquid tumors, but have had limited success to date in solid tumors. One of the main challenges of solid tumor therapies is the limited number of truly tumor-specific antigens. If an antigen isn't specific enough, there can be on-target off-tumor toxicity, or if the antigen is expressed only in a subset of the tumor cells, then you can have antigen escape. So a possible strategy that we at A2 and others have been exploring are logic gate CAR T-cell therapies that target more than one antigen. AND gates, for example, can be used when tumors express both A and B antigens to increase specificity of tumor targeting, while OR gates can be used to increase tumor cell coverage when either A or B antigen is expressed. Alternatively, NOT gates can be used to target tumor cells which express A antigen but lack B antigen to better differentiate tumor from normal cell targets. This will occur in the case when tumors either downregulate or lose expression of a B antigen. And today I'll describe our TMOD system, which implements a NOT gate. So why are we interested in NOT gates? In particular, it turns out that tumor cells are really good at losing genetic material, and heterozygous loss of genetic material called loss of heterozygosity, or LOH, occurs in about 20% of tumors. Importantly, the large majority of LOH events in cancer are believed to occur in the founding clone, which leads to islands of LOH. Because this is an irreversible event, loss of signal can then be a true tumor differentiator, which we can then exploit with a NOT gate system like TMOD. TMOD consists of two receptors, an activator and a blocker. The activator can be a typical CAR, which is directed at antigens expressed on tumor cells, like known tumor-specific antigens like mesothelin or CEA, and a LIR1 based inhibitory blocker, which targets antigens that are expressed on normal cells but are lacking on tumor cells due to LOH. And at A2, we first aim to use the polymorphic HLA class 1 as our blocker target. And in this manuscript specifically, we described a method to treat mesothelin-positive cancers. Using mesothelin TMOD, which pairs a mesothelin CAR with an HLA-A2 specific blocker for use in A2 heterozygous patients that have A2 LOH in their tumors. So across their entire body, they should be expressing HLA-A2. In some organs, there is mesothelin expression, in others, there is not. But in the tumor, there's only mesothelin being expressed. And then this difference in A2 expression then allows for an all or nothing input to the effector cells that allows for selective tumor cell killing and avoids systemic toxicity to normal tissue that expresses mesothelin. But before walking through the data, I wanted to remind everyone why mesothelin is a viable target for solid tumors, especially in the context of TMOD. Mesothelin is broadly expressed across a wide range of solid tumors, as you can see from the TCGA data here. Yet there's toxicity risk associated with current therapies due to high normal tissue expression, especially in the lung and mesothelium. Therefore, targeting mesothelin using a mesothelin CAR paired with TMOD blocking system can potentially increase the therapeutic window by blocking normal tissue targeting, minimizing this on-target off-tumor response. We benchmarked our studies against the UPenn M5 CAR, specifically because of its clinically reported activity. Unlike other previously tested mesothelin CARs in the clinic, which really had little to no activity. So in the M5 phase 1 dose escalation study, 2 out of 2 patients in the highest dose cohort had severe toxicities, which led to 1 patient death. The investigators reported that lung tissue with extensive inflammation was co-localized with normal mesothelin expression. And while it's true that several factors could have played a role, the authors suggest that the highly active M5 CAR T cells given at a high dose could have resulted in on-target off-tumor attack. And this points specifically to the fact that a safety feature like TMOD will be critical for an effective mesothelin-targeted therapy. So in the studies described in this manuscript, mesothelin TMOD is encoded in a single lentiviral vector, which includes both the LIR1 based blocker, which has an anti-HLA-A2 scFv derived from PA2.1, paired with a Gen 3 mesothelin activator. So this includes a CD8 hinge and CD28 TM and CD28 41BB, CD3 ICD. All the studies described here are done using the single vector so that you can have you can ensure coordinated expression of the activator and blocker within each transduced cell. So in developing our mesothelin TMOD system, we first wanted to identify an optimal mesothelin CAR. So we used an antibody display technology to generate novel binders against full-length mesothelin. This is described in detail in the manuscript. But the key thing is that these binders were generated, then converted into CARs and transiently expressed in Jurkat cells, which encode a NFAT luciferase reporter gene. These Jurkats were then co-cultured with HEC293 T cells with titrated mesothelin expression by mRNA transfection. And EC50s were generated from the relative response by luminescence. These EC50s were then converted to absolute molecules per cell using standard curves that we generated in-house and Keefe-Kit methodology. And what we found was that many of these novel CARs, here listed CAR 1 through 6, had EC50s around 3,000 molecules per cell, which meant that they were as sensitive, if not more sensitive, than even the M5 CAR. However, the 3,000 molecules per cell is roughly the limit of detection of this assay, so it's harder to have more detail than this. But for reference, we also included SS1, which is a widely described mesothelin CAR, as well as Atara's M912 CAR, which were much less sensitive. We next wanted to understand the quantitative features of mesothelin TMOD. So in this case, we similarly transfected Jurkat cells with either the CAR or CAR plus the blocker. So in this case, they were 2 separate plasmids used to transfect Jurkat cells. And we co-cultured them with HeLa targets, which express endogenous mesothelin with titrated levels of HLA-A2 antigen, again using A2 mRNA for transfection. So in this case, the A2 antigen is titrated, whereas the mesothelin expression is fixed at about 27,000 molecules per cell. And what you can see is that all of these CARs had a ligand-dependent, exhibited ligand-dependent inhibition by the A2 blocker. Specifically because of the combination of high sensitivity and effective functional pairing with the A2 blocker, we selected CAR3 for further study as our lead mesothelin TMOD activator. This had an IC50 of about 29,000 A2 molecules per cell. And for the purposes of this presentation, I'll just refer to CAR3 as mesothelin CAR or mesothelin TMOD when it's paired with the A2 blocker moving forward. We then map the sensitivities against normal and tumor cell mesothelin and HLA-A levels attained from either GTEx or TCGA databases to demonstrate that while normal tissues such as the lung express enough mesothelin to be vulnerable to on-target off-tumor attack. So essentially that's every tissue that's above the EC50 of the activator expresses more mesothelin than the EC50 of the activator, that in the presence of TMOD, the A2 blocker, that TMOD should be able to protect the lung and other tissues. So anything above the blocker IC50 should be able to have enough A2 expression to be protected with the blocker. Whereas in tumor targets, which express enough mesothelin but will lack A2 in the presence of A2 LOH, these should be targeted by our mesothelin TMOD platform. All other vital tissues don't necessarily express enough mesothelin to be highly vulnerable, yet they will regardless express enough A2 to be protected by mesothelin TMOD. So we next performed in vitro cytotoxicity assays with mesothelin TMOD or mesothelin CAR alone in primary T cells. We cultured these TMOD T cells with various endogenous mesothelin expressing cell lines. So this includes HeLa, SW982, and U2OS cells with varying blocker to activator ratios. So HeLa's have 27:1. We engineered in A2 here, whereas SW982s have a 20:1, or U2OS have a 2:1 beta A ratio. These endogenously express A2, so for the isogenic control, we knocked out HLA-A. And what you can see is that mesothelin TMOD, shown in the closed circles, is able to kill tumor targets just as efficiently as CAR T cells alone or M5 CAR T cells, whereas they're able to block killing of normal cell targets, match normal cell targets that express mesothelin and A2, as shown in the solid green lines. And this is true even in the case of a beta A ratio that's only about 2. So one critical requirement of a NOT gate is to be able to differentiate tumor from normal cells in multiple different contexts, including mixed cultures. So here we co-cultured A2 negative tumor targets. These are HeLa cells with A2 positive normal targets mixed at a 1:1 ratio. And here the tumor targets are shown in magenta and normal are shown in green. And what you can see is that while untransduced cells have no effect on the co-culture, this is after 48 hours now on the bottom panel, mesothelin CAR kills both tumor and normal cells indistinguishably, as they should, because both express mesothelin, whereas mesothelin TMOD only kills tumor targets, shown in magenta again, while sparing normal cells shown in green. And for more detail in the manuscript, we actually do look at several different ratios of tumor and normal cell targets and show that this holds true even at very low tumor cell targets. You still have efficient killing of the tumor cells, and at very high tumor cell targets ratios where you have efficient killing of all the tumor cells and sparing of the normal cells. Another critical requirement of a NOT gate is to be able to demonstrate persistent and also reversible killing. So here what we did is we tested mesothelin CAR only cells or mesothelin TMOD T cells in 2 serial rounds of exposure to tumor or normal cells. So TMOD T cells that were first either exposed to tumor cells or normal cells were then removed and reintroduced to fresh tumor or normal cells. So what you can see here is that TMOD T cells, which were first exposed to tumor cell targets, kill just as efficiently as CAR T cells alone, just like we saw previously, but could then be transferred onto fresh tumor cell targets, again, the closed pink magenta bar, and shown to kill just as efficiently as in round 1, similar to CAR T cells alone. In a reversible setting, these TMOD T cells could also be plated onto fresh normal cell targets and shown to block killing of normal cell targets, whereas CAR T cells, again, kill these just as efficiently. This is also true in the reverse. TMOD T cells, which block killing of normal cell targets in round 1, can then be transferred onto fresh tumor cell targets and kill just as efficiently as they do in round 1, and again can go from blocking to continued blocking. We then tested mesothelin TMOD in vivo in a dual flank xenograft model in NSG mice, where every mouse had a cell type matched mesothelin positive A2 negative tumor graft and a mesothelin positive A2 positive normal graft. Here we used MS751 cells with either HLA-A knocked out to represent the tumor graft or overexpressing A2 to represent the normal graft. And we monitored graft size over time by both caliper measurements as well as by BLI, since each graft also expressed either firefly luciferase on the tumor side or Ranella luciferase on the normal side. And when tumor grafts reached, both xenografts reached about 100 to 150 millimeters cubed in volume, mice received 20 million T cells total by IV injection. And what we observed was really clear. So on the tumor side, mesothelin TMOD, here shown in blue, was able to clear tumors just as efficiently as both mesothelin CAR alone or M5 CAR. But on the normal side, mesothelin TMOD spared killing of the normal graft and was pretty much overlapping with, xenografts treated with untransduced T cells or saline, shown in black. Whereas mesothelin CAR and M5 CAR similarly wiped out both normal and tumor grafts. And you could see this both very clearly by caliper measurement. And this was also confirmed using BLI, where you see that tumors are cleared efficiently by mesothelin TMOD, and the normal grafts are spared similarly to those treated with untransduced or saline. And this was true in 10 out of 10 mice in the mesothelin TMOD group. Interestingly, despite the robust in vivo efficacy that we observed for both mesothelin CAR, M5 CAR, or mesothelin TMOD, TMOD T cells had a relatively modest T cell expansion in the peripheral blood. So when we tracked CAR T cells in the blood over time, actually we saw at the peak response, which was about 2 weeks post T cell injection, that mesothelin TMOD T cells were about 10-fold lower in the blood compared to mesothelin CAR alone. And these were both lower than the expansion that was observed for M5 CAR. One difference for M5 compared to our CAR is also that it's in this context, a Gen 2 CAR, whereas our CAR is a Gen 3 CAR. Serum cytokines were also correlated to T cell expansion, with mesothelin TMOD and our CAR making significantly lower interferon gamma compared to M5 CAR alone. So as the name suggests, mesothelin, this TMOD system is highly modular. So we've extended the TMOD system to multiple other activators and blockers. And here I'm showing data from an in vivo study from our CEA TMOD manuscript published in Science Translational Medicine earlier this year, which pairs a CEA activator with an A2 blocker. So in a similar dual flank model here using endogenous CEA expressing H508 xenografts, CEA TMOD, and is shown in orange here, is able to clear tumor cells just as efficiently as the CEA CAR alone, whereas it's able to spare killing of the normal grafts, unlike the CAR or the benchmark. Here we use the Rosenberg's CEA A2 restricted TCR, which clears only A2 positive xenografts, but is unable to kill A2 negative xenografts. We also wanted to demonstrate that mesothelin TMOD could be extended to other blockers. If true, this would allow for the TMOD platform to extend beyond just those individuals who are heterozygous for A2. So mesothelin CAR was paired with an HLA-A03 directed blocker, HLA-B07 directed blocker, and HLA-A11 directed blocker in DRCAT cells, all of which resulted in functional blocking of the CAR in a ligand-dependent manner. Because HLA-A11 is of high interest, especially in Asian populations where the frequency of HLA-A11 is high, we further screened mesothelin CAR paired with A11 blocker in primary T cells and showed that this A11 blocker similarly blocked killing of A11 positive, mesothelin positive normal cell targets, and could kill A11 negative tumor cell targets just as efficiently as the CAR alone. Finally, we wanted to demonstrate that the TMOD system can be extended to an autologous T cell product. One can imagine that in the case of an A2 positive donor, which expresses mesothelin TMOD, that there could exist cis binding between the blocker and self A2, which would sequester the blocker and impair recognition of HLA-A2 in trans. Unlike in A2 negative donors, where the A2 blocker is free to bind to normal cells in trans, or an A2 tetramer probe as a surrogate for normal cell binding. So to address this problem, we utilized two methods that we described in the paper to specifically address the problem of endogenous HLA expression. One was B2M shRNA knockdown, and the other was B2M knockout by CRISPR to reduce HLA-A2 expression on the surface of the T cells. So here I'm highlighting the data for the ability of B2M shRNA paired with mesothelin TMOD in an A2 positive donor to downregulate surface HLA class 1, restore binding to an A2 tetramer, as can be shown here. So here, HLA-A2 tetramer binding is reduced in A2 positive donors with mesothelin TMOD, but restored in the presence of B2M shRNA. This then restores functional binding and recognition to HLA-A2 in trans and restores blocking function. So here, mesothelin TMOD in an A2 positive donor is shown in the closed circles and actually has very impaired blocking without the presence of shRNA. But when you introduce SH, which is shown in the triangles here, you can have really nice killing of the tumor cell targets, and you restore blocking of killing of the normal cell targets. So with that, I'll conclude by saying that mesothelin TMOD is highly selective, persistent, and reversible. In vivo, mesothelin TMOD displays robust tumor graft killing while sparing normal grafts. And hopefully the data I've shown shows you the modularity of the TMOD system and how it can be extended to different activator and blocker combinations to potentially cover a broad range of cancers in various patient populations. And also, so this audience is aware, A2 is currently advancing several of these TMOD constructs to the clinic. The first two being A2B530 for CEA TMOD and A2B694 for mesothelin TMOD. We've published quite a few papers on this technology recently that I hope you'll check out in addition to the key ones I've highlighted today. This molecular immunology paper that came out back in 2020 specifically detailed the extensive screening we did to end up with the LIR1 based blocker. And our most recent publication in molecular therapy oncolytics describes the crystal structure that we solved for PA2.1 binding to HLA-A2. Also want to direct you to our website, a2bio.com, where you can find our posters as well as all of our publications. We also have posters on there that we showed recently, just this last week at SITC, describing both the A2B530 and A2B694 clinical programs. And with that, I'd like to thank you for your attention and would be happy to take any questions.